Welcome to all. My topic is DNA sequence. What is DNA? DNA stands for deoxyribonucleic acid. DNA is a molecule that carries most of genetic instructions used in growth, development, and functioning and reproduction all in living organisms and many viruses. Then, structure of DNA nucleotides. Each nucleotide consists of nitrogen-containing nucleobase, either are cytokinin, guanine, adenine, or thymine, as well as sugar is called deoxyribo and phosphate group. What is DNA sequencing? The process of determining in order of base in adenine, thymine, cytosine, and guanine along in DNA strand. All the information required for the growth and development of an organism encoded in the DNA is just genome. Method of DNA sequencing method, chemical clave method, then premier extension chain transmission method, auto DNA sequencing method. First method, chemical cleavage method. The sequence DNA fragments containing up to 500 nucleotides in length. This method used double standard DNA samples. Maxim Gilbert sequencing was first widely used method for DNA sequencing. Involve modification of the phase in DNA followed by chemical phase of specific cleavage. Then stage. The double standard fragment 2 sequence is isolated and radioactively labeled at the 5 end of the with 22B. The fragment is a cut registration enzyme and thus label is removed in one end. The fragment DNA one ended label is the denatured. 4 identical samples of these end labeled DNA restriction fragments are subject to chemical cleavage as different chemical nucleotides. There are 4 specific set of chemical reactions. They selectively cut off the DNA backbone G, A plus G, G plus into C reduced. The enzymatic method. The enzymatic produce commonly referred to Kunshan method since was developed by Shankar and Go workers. Shankar sequencing is method of DNA sequencing based on the selective incorporation of chain determining deoxyribonucleic acid and DNA polymerase and in vitro DNA replication. The classical chain termination method requires a single standard DNA template, a DNA polymerase, normal deoxyribonucleic acid, trisphosphate stages. The DNA sequence is called the template. DNA is a proper single standard DNA after being polymerased vector DNA infected in the E. coli host cell release page, which contains single standard recombinant DNA, include the same sample DNA, a synthetic 5 end of labeled oligo deoxyribonucleic is used. The primer, the template DNA is hybridized the primer. The primer elongation is performed into 4 separate polymerization reaction mixers. Each mixer contains 4 deoxynucleic acid, higher concentration and lower concentration each of the 4 NTPs. The initiation of DNA synthesis by adding enzyme DNA polymerase since the enzyme cannot distinguish between normal nucleotides than alongas. Then application of DNA sequencing. The DNA sequencing may be used to determine of the sequence in the individual genus. The larger genetic regions, full chromosome or entire genes. Then forensic science. Sequencing has been used for forensic science. Identify particular individual because every individual unique is identified. To identify criminal findings, some of proof in the crime scene in form hair, nails, skin, blood sample, determine banding of the child. Medical DNA can be used to detect the gene. Different techniques like to therapy to identify two defective gene and replace them with the healthy one. Agriculture DNA sequencing is played a vital role in the field. Agriculture, the mapping the sequencing all of gene of the microorganisms have allowed agriculture to make them useful for the crop and food plants. Thank you.